Chapter 18 covers a whole nother aspect of genetics, population genetics, and it includes evolutionary genetics to some extent as well. You can study genetics at different levels. You can study genetics at the molecular level, at the level of the molecules, studying the molecules and the macromolecules like DNA and protein, and that's molecular genetics. Mendelian genetics or transmission genetics studies the expression of the phenotype in the organism. You can take, a black mouse and a white mouse and cross them and see what phenotypes you have among the offspring. That's Mendelian genetics. And then population genetics is above that level. It's at the population level, looking at how genes are transmitted or how gene frequencies or allele frequencies change in a population over time or don't change over time. So this chapter deals with some aspects of population genetics, including a model to do some calculations called the Hardy-Weinberg model or Hardy-Weinberg equation. And then it will also look at some basic concepts from evolution. So if you've had a Biology 1407 course, the material in this chapter isn't any more difficult than how it's treated in that class. If you haven't had 1407 biology, it's fine. It's not extremely difficult. So this first slide shows two populations. On the left, you have a population of humans, the human population. On the right, you have a population of ladybugs. And the rule is that organisms that belong to the same species typically exhibit variation, genetic variation and phenotypic variation. So you can see with the ladybugs, visually you can see some of the variation. It's the color of the exoskeleton here, not only this part, the orange and the red and the black and the spots, but also this white and black part is also quite different if you look at the different ones. And then in the human population, we have quite a bit of variation. But you can assume that the genes that encode things that aren't quite as visible also have that same amount of variation. Genes responsible for metabolism, cell cycle control, all kinds of things can have variations in them, and those aren't as easy to see as some of the physical traits. But the genetic variation is there, more or less, in every species. Now, the two mathematicians who came up with a model or a mathematical equation to study population genetics were Godfrey Hardy and Wilhelm Weinberg. And they didn't know each other. Godfrey Hardy was British, Wilhelm Weinberg was German. But in the same year, they published or they described a way of mathematically dealing with population genetics, and it became known as the Hardy-Weinberg Law, although Weinberg felt that his description was more complete than Hardy's, but in any case, both of their names end up attached to this law. So the Hardy-Weinberg Law is a mathematical equation used to calculate and estimate allele and genotype frequencies in a population, or actually in many populations over time, if those populations are not significantly changing. So you can say that your population is in equilibrium if the genotype and allele frequencies in your population don't change over many generations. So that's not evolving, because evolution is defined as change over time. So not changing over time means not evolving. And that's what equilibrium is defined at, a population that over many generations doesn't change significantly genetically speaking. Now, the basics of the Hardy-Weinberg Law really come back to the fundamental understanding of the Punnett square. In fact, Godfrey Hardy was friends with Reginald Punnett. And so Punnett asked Hardy to develop a mathematical model for dealing with population genetics. And Hardy basically modeled his answer after Reginald Punnett's concept. So what this shows really is something reminiscent for you. We used the Punnett square previously to show, let's say you have a heterozygous individual, big A, little a. And if that's a male individual, then half their sperm will carry a big A allele and half will carry a little a. And if you cross that individual to a female whose eggs would half of them carry a big A and half carry a little a, you have fundamentally a monohybrid cross. And so if half the sperm carry a big A and half the eggs carry a big A, then the chance that a big A sperm would meet a big A egg is 1/2 times 1/2, so that's 1/4. So the frequency of big A offspring would be 1/4. If you had the probability of a little a sperm meeting a big A egg would be a 1/2 times 1/2, and that would be 1/4. But you could also have a big A sperm meet a little a egg, and that would be a chance would be 1/4. And then there's two ways then to get a heterozygous outcome. So when you combine those together, that's a 1/2 probability of a heterozygote. And then the chance that two gametes that carry the little a would meet is also a 1/2 times 1/2, so that's a 1/4. So a 1/4 chance of getting the homozygous recessive. But what population genetics deals with is not the sperm of one individual, but the sperm of the whole population. So now when we look at this kind of Punnett square, we're saying, okay, collectively, if we take all the sperm in the population and we mix it with all the eggs in the population, and this is very theoretical, but when individuals mate randomly, potentially you have all the sperm meeting or potentially encountering all of the eggs. So if all the sperm in the population were mixed with all the eggs in the population, what would the outcomes that you would predict? So in this case, we have to consider how frequent the certain alleles are in the gene pool. So over here, when it says F with an A in parentheses, that means the frequency of allele big A. We're going to assign that as the variable P. So P represents the frequency of the big A allele in the gene pool. And Q represents the frequency of the little a allele in the gene pool. So these are allele frequencies. So how often that allele appears among the group. Then instead of saying the chance of a big A is a 1/2 and the chance of a little a is a 1/2, instead we assign the allele frequency for the whole group. Maybe 80% of the alleles in the group are the big A allele and only 20% are the little a. So instead of using a 1/2 and a 1/2 here, you say the probability of a big A sperm is 80% and the probability of a little a sperm is 20%. And among the eggs, let's say in this example, the probability of a big A egg then would be 80%. The probability of a little a egg would be 20%. So the chance in the whole population that a big A sperm and a big A egg would meet is 80% times 80%. Frequency of the dominant allele times the frequency of the dominant allele equals the frequency of getting a homozygous dominant individual as an offspring in the population. In the same way, the frequency of the little a allele, Q, is 20%, and the times Q again, so frequency of getting a little a sperm and a little a egg is 20% times 20%, which gives you Q squared, which ends up being 4%. Q squared then represents the frequency of a homozygous recessive offspring. And then the probability of getting a heterozygous offspring in the group in the next generation would be, the probability of getting a little a sperm is 20% and a big A egg is 80%, so 20% times 80% is 16%. But you could also get a big A sperm and a little a egg, and that would still be 16%. So you end up counting that both ways. 16% plus 16% is 32%. And so over here we have the frequency with which the next generation will yield a homozygous dominant individual is represented by p squared. The frequency with which you would get a homozygous recessive individual is q squared. And the frequency with which you'd get a heterozygous individual is 2 times p times q. So you may recognize some of these numbers. Another rule, other than defining these variables, is that p, the frequency of the dominant allele, plus q, the frequency of the recessive allele, has to equal 100%, assuming there are only 2 alleles for the locus. And we're going to deal only with simple scenarios where there's 2 alleles for the locus, a dominant and a recessive. I know that's overly simplistic, but assuming there's only 2 alleles, p, the frequency of the dominant allele, plus the frequency of the recessive allele has to equal 100% or 1. So we would add then p plus q equals 1. That's one of our rules. And in my example, p was 80% and q was 20%. So that has to equal 1, or if you're doing percentages, 100%. So that's an equation, a relationship that you can use later. It becomes important. And then make sure you know what p squared represents. P squared represents the frequency of homozygous dominant individuals. Q squared is the frequency of homozygous recessive individuals. And 2pq is the frequency of heterozygotes. And we used this a little bit when we did our calculations for the CODIS DNA fingerprinting test. You might remember. You might not have even realized that's what we were doing. So the criteria of equilibrium, genetic equilibrium, which is sometimes called Hardy-Weinberg equilibrium, is that you have to be studying a large population, sometimes called infinitely large, although there's no such thing truly, but a very large population that has a certain amount of genetic inertia, meaning a couple individuals live or die out of a huge number. It really won't change your allele frequencies too much, or even very much at all. There has to be random mating. And when we study something using the Hardy-Weinberg law, we're focusing only on one gene, with 2 alleles. And so it has to be random mating with regard to the trait that you're studying. It doesn't have to be random mating across the board. Obviously, all organisms have some preference. Most organisms have preferences. But with regard to whatever trait you're looking at, the mating must be random. And there can be no significant rate of mutation, migration, or natural selection. So as kind of an example, we're going to bring back a concept that we looked at previously, and that's CCR5. CCR5 is a gene that encodes a cell surface receptor protein. It's chemokine co-receptor 5 that is necessary for the HIV-1 virus to bind and to enter cells and cause infection. So you have these proteins on your cell surface, and especially your white blood cells like T cells. And one of the main ones is CD4, but CCR5 is a co-receptor. And these proteins, in an evil way, HIV uses the natural proteins on white blood cells to bind to the cell and to enter the cell, because of course HIV infects white blood cells. So I'm going to stop the lecture right now, this section of the lecture, and I'll put this link below this lecture window, and you can then go to this YouTube video and watch this. And we're looking at the concept of CCR5, a 32 base pair deletion in exon 4 of this gene confers resistance to infection. You have to be homozygous for a deletion, homozygous for a mutant allele of CCR5. Gives you resistance, a natural resistance to HIV infection. And this allele is called CCR5 delta 32. Because in genetics, the Greek letter delta, think of DEL, represents deletion.